Berislav Zlokovic. Is that how you pronounce? Okay, great. And Dr. Zlokovic would talk about the role of blood-brain barrier in neurodegeneration.
Thank you very much. And I can go to lessons learned and gaps because if we don't have time, right? Okay. So let me just try to really briefly review for you. This area is huge, so I'm not pretending I'm going to tell you very much. But I'm going to focus my talk both in animal models and humans on parasites and how they regulate the blood-brain barrier. So this is going to be my last slide, too, because we need desperately biomarkers, molecular biomarkers of function of different cell types within neurovascular unit, including endothelial cells, pericytes, astrocytes, neurons, white matter, and also we need imaging biomarkers. So we tried over the past year and a half to develop a molecular platform for about 40 biomarkers for cell-specific types within the neurovascular unit, and also developed some imaging tests both in humans and animal models, including DC-MRI, ASL-MRI, etc., and relate this to brain structural and functional changes, including diffusivity, tractography maps, but DTI-MRI, and functional load maps. So our model of small vessel disease, that I call it small vessel disease only for you this time, it's usually parasite deficient mouse model, I think is characterized by very strong blood-brain barrier breakdown. As you can see over here, this is fibrin extravasation, and that correlates with the loss of parasite coverage. More you lose parasites, you have more accumulation of fibrin. And also there is accumulation of other molecules coming principally from blood, such as thrombin, plasmin, that degrades neuronal laminin, etc. So going back to the slide that I just missed, I want to say that our model is PDGFR beta plus minus mice. That's receptor on pericytes, the green cells, which actually get signals from endothelium, from PDGF-BB, and then forms form vascular networks. So you see age-dependent progressive loss of pericytes and progressive loss of capillaries. So it's a capillary degeneration model. And as you can see is here, there are pericytes that are separated and they're dying, and also endothelial brain capillaries are dying. So they're dying together. All right, and so is that functional? So we did characterize that first in my lab by Bob Bell and Winkler when I was still in Rochester. And this was just showing by lectin angiography that we have, actually this is a perfusion, two-photon microscopy of the blood flow, by showing that at 1 month we have reduction in the blood flow already. So this is reduction in capillary length, and that goes progressively with age and correlates with the reduction of the resting cerebral blood flow, here shown by quantitative autoradiography. Very important to notice is that these changes happen very early. They happen at 1 month of age in this small vessel disease parasite deficient model that has this chronic blood-brain barrier breakdown. So all vascular changes happen very early. More recent work in this area, we were interested in CBF dysregulation following on some work of David Atwell that was published in Nature recently. And we found also in response to stimulation that we have red is capillary response and gray is arteriolar response. And we have the capillary response we also found comes a little bit before arteriolar response. But of course, both responses play an important role in functional hyperemia. But in parasite deficient mouse model, we have the reverse. Arteriolar response comes first, and we have very, very delayed capillary component. So we believe that this is the reason for low oxygen delivery. And here are oxygen tissue maps, and the blue is bad, low oxygen, and the reddish is better. So as you can see in parasite deficient mouse model, we have about over 20% vessels that have a very low oxygen, areas that have very low oxygen level, about less than 15 millimeter mercury. So these all dysregulatory vascular problems or neurovascular dysfunctions happens very early. At that point, there is no any problems with neuronal dysfunction. But we are very interested to see whether white matter is affected in this model, because white matter regions have the lowest cerebral blood flow normally and have lower capillary density compared to gray matter regions such as cortex or some other regions, hippocampus and etc. So we've done monosynaptic anterograde tracing using AAV-EGFP in barrel somatosensory cortex, and then determine the overlapping on the contralateral cortex monosynaptic pathways, also sensory motor cortex 2, and internal capsule. So as you can see, there is dramatic reduction of this connectivities through the corpus callosum very early at 2 to 4 months, and that is progressive with age. Internal capsule is less pronounced and comes a little bit later, and there are no such changes on the short pathway, so such as S1, S2. So we then wanted to study by using, DTI MRI functional maps and see how these functional maps that are here beautifully generated by our collaborator, Madeleine Doianu, also from Romania, like Zorina. And they always like these nice things to present. And so this is basically all this different tractography that we used to study pericyte-deficient mice. So how the vascular deficits influence different tracts in these mice. So the simplified version of these mice here shown just by corpus callosum, and you can see that these mice, 2 to 4 months, start losing tracts and fibers, as shown below over here. And also, of course, the older mice, they all completely lose these fibers and have a very significant loss of connectivities. What is also interesting that this white matter tracts across corpus callosum show the first reduction in the volume, while hippocampus and cortex do not show changes at that stage, but show changes only in the later stage. And this has been confirmed by calculation of a myelin axonal ratio, which is called G ratio. And as you can see, these are normal G ratios at 2 months old and 9 months old. They are parallel. And then at 2 months, this G ratio moves down, which actually suggests a loss of myelin and also number of degenerated axons. And is also confirmed at a molecular level by BMPA. And in an earlier work, we showed that at that stage, hippocampus does not show changes by, for example, a Golgi technique shown over here. But at 8 months, this is CA1 subregion of hippocampus. This pericyte-deficient mouse model, because of small vessel disease, develops loss of neurons, loss of dendrites, dendritic length, spine density, and this can also be confirmed by H&E staining. As you can see, great loss in the CA1 region. So what I showed you was white and gray matter degeneration. In a parallel work, we also showed that this model of small vessel disease developed A-beta and tau pathology when crossed with APP mice, accelerated, and also leads to tau phosphorylation and neuronal degeneration and neuronal loss, having a spiral effect. But I don't have time to talk about that. So we moved to human studies, and we use a model very similar that Gary Rosenberg talked to you about, but we extended this model to actually study in parallel several regions in the brain. So we focus on 12 or 14 regions. And what we first noticed with this model, this is, for example, a mask of hippocampus, that we have age-dependent leakiness, normal age-dependent leakiness in normal human populations. So now we have about 36 of these participants, and particularly CA1 region and dental gyrus are affected. As you can see, increase in K-trans, in permeability, while other regions are not. So white matter is fine, and all other regions are not. So then we compared this with MCI patients, which are with mild cognitive impairment, and also MS patients, which do not have cognitive impairment, but just young patients. And as you can see, there is a progressive increase in hippocampus of blood-brain barrier permeability. In MS patients, there is increase in white matter. I don't show you that, but there is no any changes in the hippocampus. And so this is now about 60 patients altogether. And so we are preparing a revision for our paper that we hope to submit in a month or so. And this relationship holds in CA1 region, delta gyrus. So these are the most prominent regions associated with cognitive decline. Now, we heard a lot about pericytes, and one model of measuring pericytes is soluble PDGFR-beta, that is pericyte marker, that we show in this group of MCI patients is increased and correlates with increase in blood-brain barrier permeability. And we also did 2 animal models, one pericyte-deficient mice and one APP mice, and we both lose pericytes, and we show also that this pericyte marker is increased. Now, molecular MRI is something that we heard from previous speakers is quite important to develop for monitoring neurovascular inflammation. And my French postdoctoral fellow, Axel Montaigne, has developed this molecular MRI method with MPO, muon particles connected with alpha VCAM. So we measure this inflammation in models of ALS, for example, and this is something that I think is important for the field. Finally, I want to mention that our previous work in ApoE, studying the effect of ApoE genotype on a blood-brain barrier, has shown that ApoE4 leads to blood-brain barrier breakdown by activation of cyclophilin A pathway, cyclophilin A MMP9 pathway, and degradation of the Thai junction. But ApoE3 or ApoE2 and murine ApoE can block this effect through LRP receptor and can block the pathway. So this is basically what we showed in mice that was published by Bob Bell, my former student, who is now in Pfizer. And we also showed that these changes happen very early. The blood-brain barrier changes with uptake of, let's say, IgG here, or thrombin and fibrin in neurons. They happen very early at 2 weeks of age, while neuronal activity, for example, cortical depolarization, is completely preserved and shows changes much later at 4 months of age. So that's, again, an example that vascular blood-brain barrier problem precede problems in neuronal function. And this has been repeated in human study. Now, in 90 patients, we first published paper, actually my graduate student published paper in JAMA Neurology, in 50 individuals showing that BBB breakdown is associated with activation of cyclophilin A and MMP9 findings in analytes in the CSF. And interestingly enough, on a larger number of samples, we also showed that E3, E4 patients have correlation with age and blood-brain barrier probability to albumin quotient, not E3, E3. And this is related to increase of MMP9 and also increase in the endothelial VCAM, for example, marker, but not, for example, astrocytes marker. And we've done all other different markers. So that's about all what I wanted to tell you. And lessons learned are that pericytes lead to BBB disruption, capillary degeneration, and blood flow reduction dysregulation. This leads to white matter lesions. Pericytes degenerate in AD. There is blood-brain barrier breakdown in aging human hippocampus and pericyte degeneration, and so implication for neurodegeneration AD is something which I think would be interesting to study. And ApoE4 BBB breakdown that is mediated by this pathway is interesting also because restoring this pathway in animal models restores the BBB and neuronal phenotype. Obviously, all about markers, I think we need to use in human studies, different markers, vascular markers, tractography, molecular markers, A-beta, and study people at risk for Alzheimer's disease, familiar, and also vascular cognitive, and similar studies in animal models. And this is a wonderful group of people that work in my lab, plus collaborations with Caltech and USC ADRC and neuroimaging group. Thank you very much.
Thank you. So we have time for some questions, quite a few actually. You don't have to buy drinks tonight, by the way.
Okay. Maybe somebody buy me.
Yes, I'll give you one of mine.
Okay.
So that's it.
Yeah. Oh, okay.
That was nice. Can you comment on the kind of the distribution of the pericytes along the cerebrovascular tree?
Yes.
And the difficulties with the fact that at the arteriolar level, you have as you transition to the capillary, you have a mixture of smooth muscle cells and pericytes, which may be difficult to kind of sort out.
Yeah.
And so, can you kind of comment on the complexity of this model?
Sure. Well, I think that's a really crucial question. I know when we first showed this in Neuron in 2010, we showed that about 70–80% of capillary wall is directly invested by pericytes in direct connection, and at that time. Then after that also, Martin Lauritsen and David Atwell showed the same, and then many labs reproduced that. So at the capillary level that are 6 microns, there is no question what it is. Now at 20 microns also there is no question it's smooth muscle cells. But what happens between 20 and 6, and where is this lovely transition happens when pericytes comes closer to his cousin, vascular smooth muscle cells, where is the transition of phenotype? I think this is a dynamic process and really depends on the precapillary sphincters, depends on many, many other roles, and we don't know much. We are trying to construct, by using STP tomography and Imari's software, the branching of all these vessels to figure out what it really is and see if we can do it using NG2 red mice, for example. But it's work in progress. But wonderful question, no definitive answer. For lower and upper part, yes. In the middle, we don't know.
Thank you.
Yeah, here. Very elegant. Two short questions. One is, can you comment if you looked at astrocytes in your parasite-deficient mice? Was there anything unusual about astrocytes?
Yes. Actually, we looked at that, and also Christer Batskell's looked. And Christer reported, we didn't report it, but we also saw the same. Parasites are important for bringing the position of astrocytes and feet to the wall. And astrocytes communicate with parasites mostly, direct contact that we show in that Nature paper and ApoE paper, but also directly with endothelium. So if you don't have parasites, this opposition doesn't exist, and there is a huge space between astrocytes and endothelial wall. So the loss of orientation of parasites and feet.
Okay. And the second question, so we know that parasite coverage increases during brain maturation, right?
Yes.
From those almost none in fetal brain and then gradual development.
Yeah.
So, and when parasite coverage is relatively low, the integrity is still in place.
Yeah.
What do you think about this?
It's a very interesting question because the coverage is not proportional to the loss of the cells. Because parasites do have what is called plasticity. So they can throw 50 processes, or they can throw 100 or 150 if necessary. So you can have loss of cells 85% or 90%, like in rat model, parasite deficient, but loss of coverage is only 70%. So it's a huge. And similar, if you have loss like 15, 20% in coverage, you can have loss of parasite 40, 50%. Amazing plasticity. We don't understand everything, but it's amazing plasticity.
Thank you.
Next question.
Sarah Tolonai from Heart, Lung, and Blood Institute. I was wondering, how does the parasite deficiency manifest itself in other organs? Have you looked at that?
Yeah, we looked in this model. We did systemic. We did analysis of complete renal function, liver function, etc. In these models that we used, that are moderate or modest parasite loss, we don't see any systemic problems. So now, why is that? Obviously, we asked. It is possible that the ratio of parasites in brain and eye is 1:1 to endothelial cells. In other cell types, if you read older literature, and I don't know the newer one, there is no much, there is a 1:10 ratio, for example, in heart, or 1:100 ratio in skeletal muscle. So it's less affected than in others. But
so we have time for one more question.
Okay, thank you.
Betsy, hi, Randy Marshall from Columbia. How are you?
Hi, very good.
Thank you for the talk. So you showed a progression of the blood flow changes that may actually precede pathological changes. Do you think this is a common instigator for both processes, or is this a cause and effect producing the clinical manifestation?
Yeah, it is very interesting question. I do believe that the blood-brain barrier breakdown at first gets leakage of things like fibrin that are vascular toxic, and then you have now degeneration, not just because of loss of parasites, but you have a positive feedback of this toxic elimination. So when this happens, it's very difficult to mount any angiogenesis vital response, so the blood flow goes down. So I personally believe that blood flow changes follow, but also according to our recent work and David Atwell's work, it's possible that they happen in parallel as well.
Thank you very much.